Today, the inoculation of the Chlorella vulgaris algae was done. This part is done 3 weeks before the continuation of the project in order to allow the algae to grow. The inoculation is the most crucial portion of the lab because if it fails, the experiment cannot proceed. An aseptic environment is essential for the success of the experiment. 12 ml of Chlorella vulgaris was mixed with 0.79 ml of glycerol and 87.2 ml of Bold's basil medium in an Erlenmeyer using a graduated pipette, a micropipette, and a graduated cylinder. I learned that a medium was necessary so that the natural habitat of the algae was recreated to provide all the resources required for its growth. At the end, the Erlenmeyer was placed on a stirring plate under the fluorescent lights that would optimize the growing of the algae. Today, the absorbance of the communal growing algae was measured to monitor its growth, since there is a correlation between the absorbance and the density of the algae. The material needed for this lab was a spectrophotometer and a micropipette. I learned why it is necessary to zero the spectrophotometer before measurements. In this case, a blank solution containing BBM and glycerol was used to calibrate the spectrophotometer in order to associate the absorbance of 0 to this solution. Consequently, when the solution containing BBM, glycerol, and C. vulgaris was measured, only the absorbance of C. vulgaris was considered. This is the two graph created at the end of the project according to the data obtained. Today, we viewed yeast cells and microalgae cells using both optical and fluorescent microscopes. The goal was to identify various organelles and observe fresh cells. In this part, I improved the method I was using to focus the microscope. Using the optical microscope at magnification 1000, it was possible to identify the nucleus, the cell wall, and the vacuole of the yeast cell. The same organelles were identified in addition to the chloroplasts for the C. vulgaris cell. Afterwards, we counted a sample of cells using a hemocytometer in order to estimate the total growth of our algae. The cell concentration of our algae was 1.24 times 10 to the power of 6 cells per ml. Today we measured the absorbance of our algae in order to calculate the number of cells per milliliters in the diluted sample using this graph. Then we washed our algae culture using a centrifuge that separated the surfactant and the algae pellet so that it would later dry and be manually crushed to extract the oil. I learned that the absorbance is at 685 nanometers because it corresponds to one of the peaks of absorbance of light by chlorophyll A. Also, for this peak, there is a graph available to calculate the number of cells of Chlorella vulgaris per milliliters of solution counter to the other peaks. Today, the quantity of free fatty acids was determined in samples of virgin oil and used oils, called the analytes, by titrating a known concentration of KOH. The presence of an acid-base indicator was necessary in order to establish the point of neutralization of the solution. The mixture turned pink when the exact volume of titrate needed was added. This step was important because it allowed us to calculate the right amount of KOH needed for the transesterification in the next lab. It was possible to observe that the percentage of fatty acids is higher in a used oil than in a virgin soybean oil, since a smaller volume of KOH was needed to neutralize the analyte. Today we perform a transesterification reaction by mixing methanol, a short-chain alcohol, and KOH, a catalyst. And then by making the mixture react with used oil and virgin soybean oil. The transesterification will convert the oil into methyl ester, the main component of biodiesel, and glycerol. Following the manipulations, I discovered that the biodiesel formed the top phase of the solution because the density of biodiesel is 0.88 gram per ml, while glycerol's density is 1.25 gram per ml. Thus, the substance with the smaller density is on top. The oil was not extracted from the algae because not a sufficient quantity would have been produced to continue the project. Combining all the students, less than 1 ml of algae oil will be yielded. Today we clean the biodiesel by decanting without disturbing the glycerol. It is essential to wash the biodiesel because there is always impurities such as free fatty acids, glycerol, methanol residues, and soap left from the transesterification. The virgin soybean oil and the used oil respectively produce 52 ml and 54.5 ml of biodiesel. The percent yield of biodiesel using virgin soybean oil was 90%, which is 8% under the expected yield. The high viscosity of the substance and the technique used to remove the biodiesel can be accountable for the difference. I learned that the conversion of used oils into biodiesel is more problematic than for the virgin oils because impurities remain in the oil since it has been cooked many times. Today we compared and verified the energy content of biodiesel fuels and regular fuels. The experimental heat combustion of ethanol, octane, petrodiesel, biodiesel from soya, and biodiesel from used oil was obtained by lighting a burner containing one of the fuels under a soda can filled with water and calculating the difference in temperature of the water and the mass difference of the burner when the temperature remains stable. I learned that octane has the greatest heat of combustion amongst all of them. The average experimental result of the class was 17 kilojoules per gram. We also observed that the biodiesel flame was much smaller than the other flames and it did not release black smoke. The combustion of biodiesel was approximately 30% efficient, which is quite good since the efficiency of car engines is 20 to 25%. Today we analyze a semi-automatic biodiesel machine made in the previous years by students. This device was built to convert soya oil into biodiesel. The compartment A is the entry of methanol and KOH. The compartment B contains water. The compartment C contains methanol and KOH. The compartment D is the entry of the oil. And finally, the compartment E contains the oil. The whole process, including the transesterification, the decantation, and washing the biodiesel with water twice, takes a total of 12 hours. This is a diagram of the continuous automatic process that Melissa Barnes and I conceived.
According to our calculations, each batch produces 50 liters of biodiesel.